Good morning, one and all. I'm Dr. B. Sridevi, lecturer in microbiology from TSWRDCW, Warangal East. So today we are going to discuss about bacterial cell wall structure. You know very well that bacteria, they belong to prokaryotic cell. So these prokaryotic cells are the primitive cells without any true nucleus. So let us see what is the organization of a prokaryotic cell. Bacteria are small and simple in structure when compared with eukaryotes. Prokaryotic bacterial cells almost always are bounded by a chemically complex cell wall. Inside this wall and separated from it by a periplasmic space lies the plasma membrane. The prokaryotic cell is a simply organized cell lacking specialized internal membrane-bound compartments known as organelles that characterize the more complex and larger eukaryotic cell. In particular, the DNA of a prokaryotic bacterial or archaeal cell is not contained within the specialized organelle called the nucleus. In contrast, eukaryotic cell does have a nucleus. The genetic material is localized in a discrete region, the nucleoid, and is not separated from the surrounding cytoplasm by membranes. Ribosomes and larger masses called inclusion bodies are scattered about in the cytoplasmic matrix. Both gram-positive and gram-negative cells can use flagella for locomotion. In addition, many cells are surrounded by a capsule or slime layer external to the cell wall. So if you see the basic structure of a prokaryotic cell, so here this is the bacterial cell, and it is having the cell wall. Some of the bacterial cells, they are having capsule external to the cell wall. So after cell wall, you can see the plasma membrane. So within the plasma membrane, this is the cytoplasm. So in this cytoplasm, we can find the nucleoid. So nucleoid, it is not a true nucleus. That means here naked DNA is there without any nuclear membrane. And in the cytoplasm, we can find ribosomes sort of 70S type and inclusion bodies. In most of the gram-negative bacteria, we can find short, thin structures which are mainly meant for adhesion. They are fimbriae or pili. Most of the motile bacteria, they are having flagella. So this is the morphology of a bacterial cell. So let us focus on the topic of bacterial cell wall. The cell wall of the bacterial cell is a strong, firm, but flexible external structure that lies just outside the plasma membrane. Except for the mycoplasmas and some archaea, most bacteria have strong walls that give them shape and protect them from osmotic lysis. Because bacteria normally exist in dilute aqueous, that is hypotonic environments, most bacterial cells would burst from the osmotic pressure exerted on their cytoplasmic membranes were it not for their cell walls. If the cell wall structure is disrupted or if its manufacture by the cell is inhibited, bacterial cells typically burst. Cell wall shape and strength is primarily due to peptidoglycan. The cell walls of many pathogens have components that contribute their pathogenicity. The wall can protect a cell from toxic substances and is the site of action of several antibiotics. The cell wall is relatively porous so that it does not greatly restrict the flow of small molecules to or from the cytoplasmic membrane. Very large molecules, however, are usually unable to pass across it. After Christian Gram developed the Gram stain in 1884, it soon became evident that bacteria could be divided into two major groups based on their response to the Gram stain procedure. Gram-positive bacteria stained purple, whereas gram-negative bacteria were colored pink or red by the technique. The gram-positive cell wall consists of a single 20 to 80 nanometer thick homogeneous peptidoglycan or murin layer lying outside the plasma membrane. In contrast, the gram-negative cell wall is quite complex. It has 2 to 7 nanometer peptidoglycan layer surrounded by a 7 to 8 nanometer thick outer membrane. Because of the thicker peptidoglycan layer, the walls of gram-positive cells are stronger than those of gram-negative bacteria. The thicker peptidoglycan layer of the gram-positive cell walls makes these bacteria relatively more resistant to desiccation than gram-negative bacteria and hence able to survive better in dry locations such as on the human skin. The outer membrane of the gram-negative cell wall makes these bacteria less susceptible than gram-positive bacteria to some antibiotics such as penicillin. See here, so this is the gram-positive bacterial cell wall and gram-negative bacterial cell wall. So outside the plasma membrane, we can find the peptidoglycan layer. So this is the plasma membrane, and outside to the plasma membrane, we can find the bacterial cell wall, which is made up of peptidoglycan. The peptidoglycan layer is thick in case of gram-positive bacteria. So this is the thicker peptidoglycan layer. In case of gram-negative bacteria, the peptidoglycan layer is thin. In case of gram-negative bacteria, outside this peptidoglycan, we can find another lipopolysaccharide layer known as outer membrane. So this outer membrane is present only in gram-negative bacteria. Frequently, a space is seen between the plasma membrane and the outer membrane of gram-negative bacteria, and sometimes a similar but smaller gap may be observed between the plasma membrane and the wall in gram-positive bacteria. This space is called the periplasmic space. Actually, the periplasmic space is the space between the outer membrane and the plasma membrane. Between the outer membrane and the plasma membrane of the gram-negative bacteria. So we can call this space as periplasmic space. So let us see the chemical composition of the bacterial cell wall. So you know very well that bacterial cell walls are made up of peptidoglycan. The cell wall of almost every bacterial cell contains peptidoglycan, which is also known as murin or mucopeptide, which is largely responsible for protecting the cell against osmotic shock. This peptidoglycan layer occurs only in bacteria and it is not found in any archaeal or eukaryotic cells. As the name implies, there are two parts to the peptidoglycan. A peptide portion, which is composed of amino acids connected by peptide linkages, and a glycan, which is the sugar portion. The glycan portion, which forms the backbone of the molecules, is composed of alternatingly repeating units of the amino sugars N-acetyl glucosamine and N-acetyl muramic acid linked to each other by beta-1,4 glycosidic bonds. Attached to most of the N-acetyl muramic acid units are short peptide chains with 4 amino acids, that is a tetrapeptide. A peptide chain of 4 alternating D-dextro and L-levo amino acids is connected to the carboxyl group of N-acetyl muramic acid. This N-acetyl muramic acid is nothing but N-acetyl glucosamine with lactic acid attached by an ether linkage. Some of the amino acids occurring in the peptide portion of the molecule are relatively unusual in biological systems. These include D-amino acids and diaminopimelic acid, which occur in peptidoglycan but not in proteins. The presence of D-amino acids protects against attack by most of the peptidases. The tetrapeptide usually includes L-alanine, D-glutamic acid, which may have hydroxyl group added in some organisms, either L-lysine or diaminopimelic acid, and D-alanine. The tetrapeptide chains are interlinked by a peptide bridge between the carboxyl group of an amino acid in one tetrapeptide chain and the amino group of an amino acid in another tetrapeptide chains. Often, the carboxyl group of the terminal D-alanine is connected directly to the amino group of diaminopimelic acid, but a peptide interbridge may be used instead. Most gram-negative cell wall peptidoglycan lacks the peptide interbridge. So this peptide interbridge is mostly present in gram-positive bacteria. The cross-linkage can occur between tetrapeptides in different chains as well as directly between adjacent tetrapeptides so that the peptidoglycan forms a strong multi-layered sheet. In fact, the peptidoglycan may be viewed as one large cross-linked molecule or sacculus. These sacs have been isolated from gram-positive bacteria and are strong enough to retain their shape and integrity, yet they are elastic and somewhat stretchable, unlike cellulose. Within the tetrapeptides, lysine occurs in most gram-positive bacteria and diaminopimelic acid occurs in all gram-negative bacteria. The greatest variation in chemical composition of the peptidoglycan occurs in the cross-linkages. In the gram-positive bacterium, example, Staphylococcus aureus, the cross-linkage is a pentapeptide composed entirely of the amino acid glycine. It is a pentaglycine cross-bridge. In gram-negative bacteria, cross-linkage almost always occur by the direct bonding of diaminopimelic acid of one chain to the terminal D-alanine of another chain. So this is the peptidoglycan monomer, so which is having alternate sugars, N-acetyl uramic acid and N-acetyl glucosamine. They are linked by beta-1,4 glycosidic bond. So to this uramic acid, so here you can see pentapeptide chain actually, but the last D-alanine will be removed at the time of cross-linkage so that we are getting only 4 peptides, 4 amino acids linked by peptide bonds after cross-linking. So initially 5 amino acids can be seen, but after cross-linking, so this D-alanine will be eliminated, D-alanine will be removed so that we are getting only 4 amino acids. First one is L-alanine, second is 2-D-glutamic acid, third is L-lysine in case of gram-positive bacteria. D-alanine, the fourth amino acid is D-alanine. So these 4 amino acids are linked to carboxyl group of N-acetyl uramic acid. So let us see the structure of peptidoglycan here. Now this sugar backbone is having N-acetyl alternating monomeric units. They are the aminated sugars, N-acetyl glucosamine, N-acetyl uramic acid, glucosamine, uramic acid, like that. So they are linked by beta-1,4 glycosidic bond. So this is a polymeric chain having alternating sugars, aminated sugars of N-acetyl glucosamine and N-acetyl uramic acid. So to uramic acid, here we can see 4 amino acids. You can see the linkage of 4 amino acids, which is a tetrapeptide. So in that, the first amino acid is L-alanine, both in case of gram-positive and gram-negative bacteria. The second amino acid is glutamic acid. Third amino acid is lysine, L-lysine in case of gram-positive bacteria. Whereas in case of gram-negative bacteria, the third amino acid is mesodiaminopimelic acid. And the fourth amino acid is D-alanine. And here, this is the cross linkage, and these chains should be cross-linked with each other in order to give the rigidity. So this cross linkage is very, very important here. Always a third amino acid of this polymeric chain is linked to fourth amino acid of another chain. Okay, so in case of gram-positive bacteria, here we can see 5 glycine molecules. It is a peptide interbridge. The linkage, the cross linkage is by means of 5 glycine molecule. 5 glycine molecules. It is a peptide interbridge. Such type of peptide interbridge cross-linking is absent in case of gram-negative bacteria. We can see the direct link. See here. So this is the chemical structure of N-acetyl muramic acid, N-acetyl glucosamine. These are the aminated sugars. Okay, this is the glucose sugar in ring form, and it is having amino group, amino acetyl group, aminated sugar. So this is the glucose ring structure. So it is having N-acetyl group, N-acetyl glucose amine. And this is N-acetyl muramic acid. See, so this is the N-acetyl glucosamine. This is the N-acetyl glucosamine. To this, lactic acid is attached. This is the lactic acid. Lactic acid and N-acetyl glucosamine, they are linked by an ether linkage. So glucosamine and lactic acid, they are linked by ether linkage. Then you are getting N-acetyl muramic acid. N-acetyl muramic acid is nothing but N-acetyl glucosamine with the lactic acid linked by means of ether bond. Now these two sugars, so here N-acetyl muramic acid and N-acetyl glucosamine, they are linked by beta-1,4 glycosidic bond. So this is the beta-1,4 glycosidic bond. Now to this muramic acid, N-acetyl muramic acid, a tetrapeptide is attached containing 4 amino acids. In that, the first amino acid is L-alanine in case of both gram-positive and gram-negative bacteria. This L-alanine, it is forming a peptide bond with the carboxylic group of N-acetyl muramic acid. Now this C double bond ONH is a peptide bond. Amino group of alanine and carboxylic group of N-acetyl muramic acid is participating in the formation of a peptide bond. And the second amino acid is D-glutamic acid. Here, the amino group of D-glutamic acid is linked to carboxylic group of L-alanine by means of a peptide bond. So this is the second amino acid in the tetrapeptide. Coming to third amino acid. So this is mesodiaminopimelic acid. It is present in gram-negative bacteria. In case of gram-positive bacteria, the third amino acid is L-lysine. So here, this amino group of mesodiaminopimelic acid is forming a peptide bond, with the carboxylic group of D-glutamic acid. And the third and the fourth amino acid is again D-alanine here. Now, this D-alanine is the amino group of D-alanine is forming a peptide bond with the carboxylic group of mesodiaminopimelic acid. And here, the carboxylic acid of D-alanine will be free here. If carboxylic acid is free, which is a polar group, The bacterial cell walls, they won't have any rigidity. So definitely this carboxylic acid of D-alanine should be linked, which is nothing but cross linkage. So this cross linkage will give the rigidity to bacterial cell wall. Now we can see the structures of chemical structure of L-lysine and mesodiamino-pimelic acid. So this is L-lysine, which is an amino acid. So it is having an extra amino group. So amino acid definitely is having a carboxylic group, amino group, and it is again having one extra amino group. So this is mesodiamino-pimelic acid. And this is the basic peptidoglycan structure in case of gram-negative bacteria. Here we can see the alternating units of aminated sugars, N-acetyl-viramic acid, N-acetyl-glucosamine, N-acetyl-viramic acid, N-acetyl-glucosamine, like that. They are linked by beta-1,4-glycosidic bond. To the muramic acid, we can see 4 amino acid, which is forming a tetrapeptide here. The first is L-alanine, second is D-glutamic acid, third is D-mesodiamino-pimelic acid, because we are just talking, this is the structure in case of gram-negative bacteria. And the fourth is D-alanine. So this is one monomeric chain here. This is another monomeric chain. And here these chains should be cross-linked. You know very well that the cross-linking will be always between the fourth amino acid of one chain and with the third amino acid of another chain. So in case of gram-negative bacteria, the carboxylic group of D-alanine is forming a direct linkage. So it can directly link to the amino group of mesodiamino-pimelic acid. You can see the direct cross-linking between D-alanine and mesodiamino-pimelic acid. Again, we can see the formation of a peptide bond between the carboxylic group of D-alanine with the amino group of mesodiamino-pimelic acid. So this is in case of gram-negative bacteria. Whereas in case of gram-positive bacteria here, so we can see the same thing, repeating monomeric units of aminated sugars, N-acetyl-viramic acid and N-acetyl-glucosamine linked by beta-1,4-glycosidic bond, just like your gram-negative bacteria. And to the muramic acid, again we can see the tetrapeptide unit here. So first is L-alanine. This L-alanine is forming a peptide bond with the carboxylic group of N-acetyl-viramic acid. Second one is D-glutamic acid. The second carboxylic group gets amidated during the cell wall synthesis in order to give a more rigidity here. And the third amino acid is L-lysine. In case of gram-negative bacteria, the third is mesodiamino-pimelic acid. Whereas in case of gram-positive bacteria, the third amino acid is L-lysine. And the fourth is D-alanine. You know very well that always the cross-linking will be between the fourth amino acid of one chain with the third amino acid of another chain. That means the carboxylic group of alanine should form a peptide bond with the amino group of another amino acid in the another chain. But here this D-alanine is linked to L-lysine by means of a peptide interbridge. So this type of peptide interbridge is absent in case of gram-negative bacteria, where you can see the direct cross-linking. But in case of gram-positive bacteria, we can see a peptide interbridge. So this peptide interbridge, it consists of 5 glycine molecules. D-alanine is linked to L-lysine by means of a pentaglycine cross-bridge. So now this pentaglycine cross-bridge is nothing but it is a peptide interbridge. And this is the main sensitive region for penicillin antibiotic. So that's why mostly gram-positive bacteria are susceptible to penicillin. So such this peptide interbridge is absent in case of gram-negative bacteria. So that's why gram-negative bacteria they can resist penicillin. And we can see the structure peptidoglycan. So this part represents, this sugar part represents glycan part, and this amino part, amino region, tetrapeptide region represents a peptide region and glycan region, peptidoglycan. So here in acetyl glucosamine, glucose N amino group, this is the acetyl group, CH3 C double bond. Well, this is the acetyl group, this is the amino group, this is the condensed ring structure of glucose. Glucose N, acetyl, glucose, amine. And this is N-acetyl muramic acid. They are linked by beta 1,4 glycosidic bond. So this is like that, these alternating sugars are linked by beta 1,4 glycosidic bonds. This represents the glycan part of peptidoglycan. And this is the peptide part. This is the peptide part, which contains tetrapeptides, which are linked by cross-linking, peptide cross-linkages. This is the structure of gram-positive bacteria. L-alanine is linked to muramic acid, D-glutamic acid. Third is lysine, L-lysine. Fourth is D-alanine. Now we can see the cross-linking between the third amino acid of this chain and the fourth amino acid of another chain. So this cross linkage will be by means of a peptide cross linkage. So this is the penicillin sensitive site, pentaglycine cross linkage. So this is the basic structure of gram-positive peptidoglycan. As this pentaglycine cross bridge is absent in case of gram-negative bacteria can resist penicillin. You can see this again another diagrammatic representation, N-acetyl glucosamine and muramic acid. So alternating sugars to the muramic acid, 4 amino acids are there, 1, 2, 3, 4. Like this 4 tetrapeptides are there. And you can see the cross linkage between the tetrapeptides. See, the linkage will be between the third amino acid and the fourth amino acid. You can see here. Third amino acid is linked to fourth amino acid. In case of gram-negative, this linkage will be a direct linkage. In case of gram-positive bacteria, this linkage will be by means of pentaglycine molecules, pentaglycine interbridge. So this is about the basic structure of a bacterial cell wall. So I'm just focusing on peptidoglycan structure. In the next class, we will be discussing the differences between gram-positive cell wall and gram-negative cell wall. So let us have a glance on the summary of this topic. The vast majority of the domain bacteria have a rigid cell wall because the cell wall is rigid because of the presence of peptidoglycan. And the peptidoglycan cell wall surrounds the cytoplasmic membrane, you know very well, and it just protects the cell from osmotic lysis. Peptidoglycan is composed of interlocking chains of building blocks called peptidoglycan monomers. You know that peptidoglycan monomers are having alternating units of N-acetyl glucosamine and N-acetyl muramic acid and has a cross-linked short peptide chains, some of which have unusual amino acids such as D-alanine. Only peptidoglycan is having unusual amino acids like dextroform, D-alanine. The crosslinks provide the needed structural support of the wall. The tetrapeptide side chain is composed of alternating D and L amino acids, and the mesodiaminopimelic acid is present in gram-negative bacteria. So this is about the basic structure of bacterial cell wall, peptidoglycan. So just see the assignment here. You just go through the assignment and just read the content and submit the assignment in the next class. Thank you.